Okay. All right. So we left off on Friday addressing this question here. What does it mean to be human? What does it mean to be human? And I think all we had on Friday were some philosophical descriptions of what it means to be human. And they all revolved around this idea of that we are made in the image of God, which I think is a perfectly... I didn't even want to say satisfactory because that sounds pejorative. It is a perfect way to answer that question. What does it mean to be human? It means to be a creature, a creation made in the image of God. Now, you may have some questions about, what does that mean? And I think some of us started giving qualities, right? It means that we are capable of reasoning, right? Or it means that we have a high level of intelligence or various other capabilities that we have. I want to caution you against using that to describe what it means to be human. Saying we are made in the image of God is a perfect way to answer what it means to be human philosophically. Using capacities that we have, I don't know if that's the greatest way to answer those questions, because then what it forces you to do is if somebody is incapable of doing that, and that's what you use to define what it means to be human, now you would have to say, by my definition of what it means to be human, this individual is not human. Okay? So I want to caution you against doing that. I mean, you can say that made in the image of God, and then you can say, which includes some of these capacities, but be careful defining what it means to be human based solely on those capacities. Okay? What about biologically? What does it mean to be human biologically? And I know we haven't talked a great deal about what is a species, how do you define a species. Those conversations will come next week. But yeah, Levi. Okay, that we stand upright. I love it. I love it. But birds also stand upright, as do kangaroos. Unless a kangaroo is walking, then they crawl. But when they're hopping, they're upright as well. But there's certainly, you can see at least within primates, there's something very meaningful about that. If you apply it to, if you say what it means to be human is an upright walking primate, now we have something that's very, very intriguing, and I think probably very biologically meaningful. An upright walking primate. Yeah, any other ideas? Okay, let's talk about traditional systematics, and then we'll talk about an evolutionary approach to this question. So traditionally, chimpanzees, gorillas, orangutans, the great apes, they were placed into a family called Pongody, and then humans and other human-like species, other species like our species, like Homo sapiens, were placed in a separate family, Hominidae. Okay, so we've got Pongody, the great apes, and some extinct forms. Hominidae, our species, and some extinct forms, two separate families. However, with a push to make everything represent the evolutionary trajectory, which is basically what we've been doing with these phylogenies, right? And we're saying, okay, well, taxa that don't represent an evolutionary trajectory, those are not valuable taxa anymore, right? Last week, we talked about how reptiles... that taxon is only valuable if you make it more general with respect to some of the fossil forms, or more specialized, sorry, not more general, more specialized, so not include some of like the synapsids, because those can't be considered reptiles, then mammals have to be considered reptiles, right? Right? We can't call the synapsids, the early synapsid, mammal-like reptiles because they're not reptiles, right? Reptiles gets less general in some of our fossil forms, but then also includes birds, right? And the drive to do that is to make every taxon represent an evolutionary trajectory. Well, this is unsatisfactory in that mindset because the idea is the ancestor that humans share with chimpanzees would have likely been classified in the family Pongody. Does that make sense? So, like, the idea is not that humans evolved from chimpanzees, rather that humans and chimpanzees and bonobos as well share an ancestor, and that ancestor would likely have been classified as an ape, okay, in family Pongody. Well, that makes this family Pongody not a taxon that represents evolutionary trajectory. So we eliminated that family Pongidie and put all of the great apes into our family, Hominidae. And so now when you look at it and you look up family Hominidae, it includes chimpanzees, gorillas, orangutans, bonobos, a number of extinct ape species are all inside of what has traditionally been the human family. The human family. All right? Does it make sense why you would get rid of Pongidie? Okay. I hope so. If it doesn't, you know, we can talk about this further. Now, doing this creates an interesting issue with the phylogeny. Okay? So when you do a phylogeny of now Hominidae, including chimpanzees, bonobos, gorillas, orangutans, number of extinct, like Paranthropus, I kept saying extinct forms but wasn't giving you any examples, Paranthropus and many of our Australopithecines. Anyways, we can talk more about those in a bit. But when you include all of these groups, you find a number of polytomies. And I'll show you a phylogeny that will show you some of these, not all of these, but some of these. And again, what do polytomies indicate? The history is unresolved, right? We do not know how these organisms are related exactly. Or, if your view of origins holds that they are separate creations, what those polytomies emphasize is that you are trying to group organisms together that ought not to be grouped together, at least as far as an evolutionary history is concerned. Okay? Even just within our genus, the genus Homo, there are several polytomies, which on the surface would make you suggest that maybe we're trying to group species together that aren't part of the same kind, right? And that's why there are polytomies. And then so it's like, okay, that's awesome, right? Adam and Eve, they're our species, and everything, no matter how human-like, is certainly not our kind, right? Do you understand, like, the desire to do that with this polytomy? We're like, there's a polytomy just within our genus, so that's like, man, our species, separate creation, doesn't even connect with any other species in our genus. Okay? Does that make sense? All right. Here's an issue. One, we find several sites. Dmanisi is a great example of this, that has multiple species of genus Homo in the same place. And it's like, not just in the same geographical area, but in the same archaeological site. Where it's like, this was representative of a single society, and there are multiple species in this genus in the same society. We're like, now this is a little bit more interesting, because the polytomy makes us want to say that these are all separate because the relationships are unresolved, and yet multiple species of this genus are living together in the same society. And it's like, is it our species putting other species in this genus in cages to watch them and to look at them? I mean, we do that now, right? There are no living species within our genus besides our species, but we put animals in cages, right? Is it that? Not likely in an agrarian society, right? Where they're, starting, to really use agriculture for the first time. It seems absurd that that's something that, anyways. On top of this, in our genome, we have Neanderthal genes. So we have Neanderthal skeletons that were preserved well enough that we can get their genome. We have a relatively complete Neanderthal genome. And then obviously we have a fully complete genome of our species, right? They mapped the human genome in the early 2000s. Now they're on an effort, I think, to do the 1 million genomes project, to get 1 million genomes matched or mapped. Okay, and so we have a copy of our genome. We have an almost complete Neanderthal genome, and within our species, there are Neanderthal genes. And the only way to explain that is that our species and Neanderthals are capable of forming offspring together and did. And we are descendants of those offspring. Right? So now it's like, okay, well, we've got multiple species living in the same society, and now we have multiple species forming offspring together. And we're like, okay, well now it looks like that the humankind is more than just our species. And then we can get into discussions about what does it even mean to be a species, because the biological species concept suggests that if two populations are capable of interbreeding, they are not separate species, right? And so is even classifying them as Neanderthals, is that even valuable? Or are we all just a single species? But it's interesting, okay? So it looks like that the humankind is more than just our species, and yet we need to figure out how this all works together. So one more piece of information, then I'll show you the polytomies, and we'll kind of, I don't know, put a bow on this little project. So Dr. Francis and Dr. Wood published a number of papers, a couple of book chapters in the book What Happened in the Garden and in other books to show that there is a definite discontinuity between our genus Homo, and possibly one Australopithecus species, although many people that study this human evolution suggest that this could just as easily be labeled HomoSediba rather than Australopithecus sediba. It looks more like a species within our genus than it does like species in this genus. This genus includes Lucy. Y'all familiar with the Lucy skeleton? That's Australopithecus afarensis. Australopithecus sediba is way more like us than it is like Australopithecus afarensis, like Lucy. Anyways, okay? So looking that there's definitely a discontinuity between our genus and possibly this species and all other members of family Hominidae. That there's a definite discontinuity between our genus and possibly this one that probably should be in our genus and all other genera. Definite discontinuity between humans and chimpanzees, definite discontinuity between humans and gorillas, humans and orangutans, humans and all of these ancestral ape species, and so that whenever a new specimen is formed, it always falls into it's either human or it's not. It's not blurring the lines. The lines are not blurred when you actually come at this not assuming that they have shared ancestry. If you come at it assuming there's shared ancestry, you can find similarities, right? We can find similarities between humans and chimpanzees. Would you agree? Yeah. Okay. If you come at it assuming that they're related, you're going to emphasize those similarities. If you don't come at it assuming they're related, you see definite evidence of discontinuity, where they are not linked together biologically. They're more different than they are similar. However, it's hard to argue that there aren't multiple species within the humankind. So then you have questions, okay, well, where did these species come from? And Dr. Francis and Dr. Wood present an argument, maybe an explanation that tends to be the common creationist explanation. That what you're looking at are morphological differences that are the result of partial reproductive isolation. In the same way we have evidence of this now with living human groups. Right? We have living human groups that are partially reproductively isolated or entirely reproductively isolated, and sometimes have some unusual morphological features. Like there are populations of Amish groups that have a really high prevalence of polydactyly and a very rare type of dwarfism. But it's because they're relatively reproductively isolated. And so if all you had was one of those skeletons and a skeleton of our species, you would probably classify them as separate species, and yet it's obvious that they're the same species. Okay? So our challenge is we don't have any living representatives of these other species. And so it makes the differences, I don't know, maybe seem a little bit more pronounced. Okay? All right. So all this to say, here, let me show you the phylogeny and some of this, and then we'll put our bow on it. Okay, so here is family Hominidae. Notice it's including all of the apes now are inside of this grouping here. Pongidae, Gorillidae, Panini, the chimpanzee, and the bonobo. And then so here's a polytomy here, one branch splitting into 1, 2, 3. Here's a polytomy, one branch splitting into 1, 2, 3. Here's a polytomy, one branch splitting into 1, 2, 3. Here's a polytomy, one branch splitting into 4, one branch splitting into 3, one branch splitting into 4, one branch splitting into 4. Lots and lots and lots of polytomies, which again is an indication of what? That these relationships are unresolved, or that we're trying to link species that should not be linked together. Okay? And so when we look to this and we're like, okay, there's a polytomy here grouping our species with Neanderthals, you could interpret it that way, that we're trying to link species that should not be grouped together, and yet we have Neanderthal genes. Okay? So here, I think it's just a case where it's unresolved, because we have very few specimens that we classified as these species, and we have no living forms. If we had living forms, I think what's interesting is the amount of variation within this genus. If you take all the species within our genus, the amount of variation among all those species is less than the amount of variation within individuals of our living species. Now, there are definitely skeletal features that we're able to say, okay, this is Neanderthal, this is, Homo erectus, this is Homo sapiens, but the amount of total variation is less than what there is in living members of our species. Okay. Here's a skull here's Lucy's skull, Australopithecus afarensis. The dark parts are the actual parts of the skull we have. Everything else has been assumed from, how skulls tend to work. So it's a small percentage of the skull, and there's only, I think, two. There's Lucy, which is the most complete skeleton, and then there's a smaller one. And then here is our species. And then here's the total skeleton we have of Australopithecus afarensis, of Lucy. Notice a lot of pieces are missing. Here's Homo erectus, much more complete idea of what the organism actually looked like. And then here's, of course, an artist's representation of Neanderthals. But yeah, so to tie a bow on this, it's obvious the distinction between human and not human. Okay, that's a very obvious distinction, no matter what characteristics you use. If you only use living species, it's very easy to say human versus not human. No matter how many of the fossil forms you include, it's very easy to say human versus not human. But another thing is it's very hard to argue that the humankind is only Homo sapiens. It looks like we have some real meaningful morphological differences, and yet we all built societies together. So it's like, what are we even seeing here if it's anything other than just some unusual morphological features because of maybe partial reproductive isolation and then coming back together. And so you could argue that Neanderthals never went extinct, but instead they merged with our species. And our species is a hybrid between Homo sapiens and Homo neanderthalensis, mostly Homo sapiens and some neanderthalensis. If you are of European descent, you are probably closer to the 7% Neanderthal. If you don't have a great deal of European descent, you might be closer to 4, or maybe even a little bit less if you're lucky. But there's a pretty good chance at least 4% of your genome is Neanderthal. And that's, I mean, the only thing you could do with that is to really say that both those species are part of the same created kind, and you could argue that maybe we shouldn't consider them different species. But yeah, Chris.
How do we know what the Neanderthal genome is, like compared to like?
Yeah, because we have, that's good. We have a Neanderthal skeleton preserved well enough that we can isolate DNA from its cells and actually construct a Neanderthal genome. Yeah. And then compare that to like a really, the oldest skeleton that we have of our species and get that genome, and then compare the genome of a modern human to our own species but an older specimen and Neanderthal, and then you can see where the genes actually came from. That the genes came from the Neanderthal genome and not the oldest representation we have of our genome. Yeah, that's a good question, though. Yeah. So this is where it gets fun, because then you start to think like, okay, if there are multiple species, I mean, again, we can argue that it's irrelevant to even consider them different species, but there are some real morphological differences. Then you get to, okay, well, Adam and Eve, obviously historical figures, right? The Bible makes it very clear. Adam and Eve are historical figures. Paul builds his entire theology of salvation upon Adam being a literal historical figure, right? and Christ being the second Adam. Okay? And then so the question is, did they look more like modern human or some, fossil human, right? Did they look more like a Neanderthal or did they look more like our species? Okay? And then again, we can argue, like, what does it even mean to classify them as separate species? But I think there's a lot of evidence to suggest that every species within the genus Homo is all part of the original humankind, and that the different forms are the result of partial reproductive isolation and getting some unusual morphological features. Okay? Like some aboriginal groups with like unusually short stature. Right? Okay? Any questions? Okay, we're going to do a lecture break. And what I want you to do for this lecture break is this. I want you to get together with those around you, and I want you to basically take, if you find somebody that has the slides available, look at that polytomy that includes basically all hominids. Focus just on all of the individuals of our genus, plus Australopithecus sediba, which is going to be with Australopithecines. And I want you to come up with a phylogeny that includes all of the species of the genus Homo and then Australopithecus sediba. Because if these are all in one created kind, there should be a phylogeny to represent that group. Okay? If they're all a single created kind, you should be able to get a phylogeny. And if the relationships are unresolved, it might be because, bless you, we don't have enough specimens, or we're overemphasizing some characteristics and ignoring some others. All right? So take a couple minutes, maybe 3, take just the species in the genus Homo plus Australopithecus sediba and put together a single phylogeny to include those forms. Ignore all other forms. All other forms. All right? Does this make sense? It's just, putting this out there, this might be a helpful thing to do. Helpful thing to know how to do. All right? Take a couple minutes, starting now.
we do as he does.
Is that spelled correctly?
No, yeah, I think so.
Wow, this marker is very stinky. All right. So, Levi, you mentioned something when I said, let's get a biological definition of what it means to be human. And what did you say?
So they stand upright.
Yeah, they stand upright. Let's say walk upright, but stand upright is fine as well. The problem with stand upright is a lot of animals will stand upright, but they don't. It's not a good way for them to move, right? A chimpanzee can stand upright, but when it really needs to boogie, it doesn't walk upright. Okay, I mean they can walk upright a little bit, but they're about as good at walking upright as I mean they're a little bit better than a dog, but not a whole lot. Okay, so let's go walk upright. Okay, and this can be something that sets humans apart from the other groups. Now we need to split this and to start, now let's focus on certain features. Okay, and so if you're going to split this branch into two branches, right, you're going to at least want to include something for this branch that we're like, okay, all of the branches that come from this branch have this feature not found in this branch. Okay? And so what we would get into is, I'm just going to write generally because you don't need to know the specifics, but some skeletal features. And then over here would be something that lacks those skeletal features, and this would be Australopithecus sediba. And again, maybe you could make an argument that this should be HomoSediba, right, in our genus. But anyways, now we've got some skeletal features that separate the organisms that have traditionally been put into the same genus as us and others. And then we'll split this further. And again, we can include all of these various species, or we could simplify this a little bit, which I think is helpful when we work on something like this. And the three most popular, if you will, species within our genus. Okay, the three most popular species within our genus would be first Homo erectus, and then two other species which we will separate as obvious tool usage. You're like, is that a biological feature? It sure is. It sure is. Gosh, this marker smells too bad. I can't do it. And then we've got Homo neanderthalensis. Ran out of space, sorry. And then our species, Homo sapiens. And this is a fairly simple phylogeny. I know it leaves out a number of species, but a relatively simple phylogeny. Yeah, Allison.
So Homo erectus didn't use tools, so there's no evidence that they
There's not obvious usage. Again, the Dmanisi site, I think many of those species would be classified as Homo erectus, and it's a pretty well-developed society, but there's some debate there. It's very obvious that Homo neanderthalensis used tools, built complex societies, buried their dead, hunted in groups, painted caverns, right? Used art and made beautiful things. Homo erectus is a little bit more complicated, but again, this Dmanisi site, which is in Europe, most of the specimens there would probably be classified as Homo erectus. And it's a society, so it's a little bit less obvious. Okay, this is something that's helpful. And then what I want to do is I want to talk about this real quick before we transition to chapter 19. How would we know, if we don't have any living specimens of this, how do we know that they would walk upright? Okay, by their skeleton. What kind of skeletal features do you think would indicate that they walked upright?
Long legs and short arms.
Long legs and short arms. That absolutely would be one indication. At least saying arms shorter than the legs, right? If your arms are shorter than the legs, it's a little bit difficult to walk on all fours, right? Now your legs, now there's some variation in that, because if you measure a dog from its hip to its toe versus a dog from its shoulder to its toe, the shoulder to its toe is a little bit shorter, but that's because of how... Anyways, but a significant difference is usually an indication that they are not walking on all fours, unless you've got... you know how it is if you try to walk on all fours. You either got to use your knees or your head's just like dragging on the floor, right? You got some weird... It just doesn't work well. It doesn't work well. Yeah, Micah?
Hip and neck structures.
Hip and neck structures, absolutely. So, true or false, our bodies are pretty heavy. For some of us, it's more true than others. But for everybody, our bodies are pretty heavy, right? I think the average American female weighs something like 135 pounds. The average American male weighs like 165 to 170 pounds. It's a pretty decent amount of weight sitting on your hips. And so if you walk upright, your hips are structured like a bowl. Like a bowl to help displace that weight, so not all of the weight is sitting on a particular part of it, but rather it's more shaped like a bowl and more evenly distributes the weight being supported by the pelvis. This is kind of cool, right? So you can look at, say if you look at a human pelvis versus like a chimpanzee pelvis, a human pelvis is much more bowl-like. It rather than sitting like this, to where the back of the pelvis is pretty close anteriorly to the front of the pelvis, it's displaced backwards and kind of is shaped, again, more like a bowl. And then you said neck structure. What do you mean by that?
Well, your head's got to be, if you're on all fours, like a human's head is straight up and down.
Yeah, so your head's got to sit on top of your vertebral column rather than in front of it. Right? Organisms that walk on all fours, the vertebral column enters into the back of the skull. Whereas organisms that walk upright, the vertebral column enters in on the bottom of the skull. And so when you look at Australopithecus sediba, it's very clear that this species walked upright, although so did the other Australopithecines. Lucy probably walked upright because the opening is on the bottom of the skull. And although we don't have a great skull for Australopithecus afarensis, but it seems that it came. So maybe walked upright isn't the greatest classification because it looks like we do have some species that are grouping apart from humans that maybe also walked upright. But it's very clear that Australopithecus sediba, that our species, that all of the other species within our genus walked upright. Allison?
Oh, I just was going to ask, so then for the apes that walk on all fours,
Yes.
then are their shoulders the more weight-bearing part of their body, or?
Yeah, I mean, it's a little bit different. So the limbs support the weight more heavily, displaced over all four limbs rather than just on two, right? If you walk upright, all of your weight has to be carried by two limbs. And so you want to try to evenly distribute as much of that weight as possible. And the only way to do that is to allow the pelvis to shoulder that weight and then distribute it evenly to the femurs. If you walk on all fours, your center of mass is not centered right on your pelvis like it is if you walk upright. Your center of mass is more like towards the shoulders, but it's in between the shoulder girdle and the pelvic girdle is where the center of mass is. It's closer to the shoulder girdle, but the weight is spread evenly, relatively evenly over all four limbs. Although with a lot of the primates that walk on all fours, the forelimbs are much stronger than the hind limbs and do bear more of the weight. Yeah, but it's not like a hard, fast rule because it can vary a little bit. All right? Is this all right? Does this make sense? Do you feel like you could communicate a little bit about what it means to be human biologically? Okay. Because here's where it gets really fun. You can start adding things in like... sorry, I'm just going to confuse you, but I'm going to do it anyways. You can start doing things like, if you want to emphasize, and I told you, be careful emphasizing like capacities, right? Like intelligence or reasoning or, really high levels of learned behavior, right? Because then it's like, okay, but if we find examples of this in other animals, now we have an interesting conundrum, right? Do they also have some of the image of God? Ethan, on Friday, you talked about the research of morality in monkeys, and then you sent me the paper, and it seems to be, they seem to have made a very convincing argument that the monkeys, if they know their partner is getting a better reward than they are, they will not work as hard as the partner. Right? If they know from the beginning that their they have to do a shared task, and they know what the reward is that they're getting compared to their partner, they won't work as hard if their partner's getting a better reward. If they're getting the same reward, even if it's not a better reward, right? They did cucumbers and grapes. This one, I don't know if I was as convinced over this argument, but it seems to be that even if you're giving them the lesser of the two rewards, I don't know if you know this or not, but grapes taste better than cucumbers, right? I mean, you may disagree, but you're wrong. And so if they and their partner are both getting cucumbers, they'll work as hard as their partner will. But if they're getting a cucumber and their partner's getting a grape, they won't work as hard as their partner because they know their partner's getting a better reward. They'll let them work harder. And so it's like, are they understanding this concept of justice, right? Where it's like, I'm not getting as good a reward, so I'm not going to work as hard for it. And I don't know if I was super convinced about that argument anyways. But it seems to be like that they're reasoning, right? It's not just associative behavior. They're not associating like, hey, when you see a grape, you're going to get a grape. No, they seem to understand when the grape is meant for them versus their partner. And it's not just like, when we got associative learning, we have visual learning where like they're associating. No, it seems like they're actually reasoning. And then so it's like, man, if we put reasoning as what it means to be human, now we've got kind of an interesting issue here because it seems that some animals are capable of reasoning through. Now, they did make it a point that they don't really understand justice. Like, so the title of the article and the abstract, it's like, oh man, this concept of justice. Because if they don't seem to mind getting a better reward than what their partner is. Right? They don't seem to mind that, and that's a true understanding of justice, right? Because the other one, it's really selfishness, right? If somebody's getting something that you're not getting, you're not going to work as hard for that. That's not really an understanding, at least I would argue, that's not really an understanding of justice. That's more like an understanding of, like, hey, they're getting something better than I am, right? But to truly understand justice, I think you could argue that you should feel that something feels wrong about getting something better than your partner that's doing the same amount of work, right? Anyways.
So, in keeping with all of that, how do we understand like apes that can use sign language or have been taught? Like, are they actually forming sentences on their own, or are they just
with limited capacity? Yes, it is more than just associative learning. It is more than just like, you know, like a rat. You can train a rat to pull a lever to get a reward, right? And that's associative learning. It's associating some action with getting some reward, or visual learning. But no, I mean, it seems like they are capable of again, critical thinking. Like, you know, thinking about how do I want to communicate this and actually communicating that. Now, within limits, their communication is certainly not sophisticated. But yeah, again, I mean, so it's, and that's one reason I think why we should be hesitant to describe what it means to be human as far as our capacities. Because that's one issue. If you have animals with those capacities, now it's like, do they have some of the image of God? And then if humans lack those capacities, do they not have the image of God? Right? You've got two issues there. And so I think it's better to just be more general and say humans, what it means to be human is to be created in the image of God, and then either provide an explanation for what that means, which I don't think you should do it as capacities, but rather position, right? That what it means to be made in the image of God is a position of authority, right? Whether you can exercise that authority or not, completely different question than whether or not you have certain capacities, right? Because sometimes your ability to exercise authority is independent of your capacities, right? So anyways, just some thoughts, some philosophical thoughts for a biological question. Any other thoughts or concerns, things you want to share? All right. I don't know if you have big issues or little issues with the idea that the humankind is more than just our species. If you have big issues with that, I don't know what to do with it other than just to tell you that it's still very clear what's human and what's not human. Okay? All right. Chapter 19. I know it feels a little weird to like get to chapter 29. We started, I think, at chapter 22, or was it 21? 21 or 22, and we've been working our way up. We get to chapter 29, then all of a sudden, it's boop, back to chapter 19. But this is how it works, okay? It doesn't have to work that way, but I made it work that way, so you're welcome, I suppose. So chapter 19 is basically, we're going to now talk about the mechanisms of evolution. So we've talked about these various groups and answered the question, if they're related, then how, right? That's what every chapter has done so far, is basically, if all bacteria came from a single bacteria ancestor, how would that work? Right? I don't know if you know, if you recognize that that's what the chapter was all about, but that's really what the chapter is all about. And then chapter 29, if vertebrates are all related, then how, right? That's what 29 is all about. Chapter 19 is giving us the mechanisms behind how populations change through time. Basically providing the mechanisms by which you can get these branching events. Okay? All right. So chapter 19. We'll talk about what is genetic structuring. How does the Hardy-Weinberg equilibrium work in maintaining genetic structuring? And yeah, there's no way we're going to get through the first three, so we'll just start moving through some of these. We have 11 minutes left of class, by the way. So I know you're like, wow, it went so fast. It's like the greatest experience of my life. So what is genetic structuring? What is genetic structuring? First of all, you need to know what a population is. A population is a collection of all of the individuals living in the same place at the same time. And time is an important part to keep in there when you're talking about fossil forms. Fossil forms. That you need to be thinking in terms of time, and so you should be thinking in terms of a single layer in the fossil. Anyways, all of the individuals living in the same place at the same time. All right. So the dominant paradigm in science is the neo-Darwinian synthesis, or sometimes called the modern synthesis. That's the dominant paradigm of explaining the origins of life as we know it. The origins of life, not the origins of, actually, that's a horrible thing to say. Not explaining the origins of life, but explaining the origins of the diversity of life. That's a better way to say it. The neo-Darwinian synthesis does not really address where did life begin? How did you get life from non-life? It's just saying, assuming you already have life, how does that diversify into all of the forms we have now? Okay? So the neo-Darwinian synthesis is a fusion of Darwin's original thinking along with Alfred Russel Wallace's thinkings about evolution by natural selection, and then a Mendelian understanding of inheritance. Okay? So this is the modern synthesis. And so here's a quick description of what this means. So you've got characters, and this, the example is given of eye color, that they come in different traits. Okay, so you've got a character such as eye color that comes in different traits or different forms or different flavors, if you will. Blue and green. Do blue and green eyes taste differently? Probably, but I don't know. Have you ever eaten an animal's eyeball? It's a different experience, especially if you eat it from a marine fish, because it just tastes like the ocean. It's an interesting experience. Anyways, sorry. So some characters are controlled by a single gene, and if that's the case, then each trait is a different allele for that gene. Okay? So if a character is controlled by a single gene, so let's say hypothetically, eye color was controlled by a single gene, then every trait would be a different allele or a different form of that gene. Now, human eye color is controlled by at least 16 different genes. So it's not that simple. It's not that simple. Which is why there are different shades of blue and different shades of green and different shades of brown and different varieties of hazel, because it's more complicated than just a single gene. So I don't know if you ever wondered, man, I have blue eyes. Neither of my parents have blue eyes. Are they really my parents? See, both of my parents have blue eyes and I have blue eyes, but neither one of my siblings did, and I wondered. But then it's very easy to accomplish that and to make that happen. Plus, my brother and I have both done 23andMe, and I know he's actually my brother, which is both encouraging and discouraging at the same time. If you're listening to this, Chris, I love you. All right. So we can measure the frequencies of each allele in the total population, and we call that the gene pool. So again, assuming we have a single character, single gene situation, right? eye colors, let's assume for a minute eye color is controlled by a single gene. Every version of that gene is a different allele. Then we can get the frequencies of each of those alleles in the entire population, right? And it would be relatively easy to do, of course, depending on the size of the population and, you know, how many different forms of that gene that there were. Some human characters are controlled by a single gene, like the hitchhiker's thumb. Impressive, right? That is 90 degrees. That's controlled by a single gene. A widow's peak, controlled by... So there are some characters that are controlled by a single gene, but it's not usually that simple, okay? Anyways, but assuming it is, we can figure out the frequencies of each allele in the population. Now, the genetic structuring for a population then represents how these allele frequencies contribute to the genotype, keeping in mind that every individual is diploid, right? Meaning they have 2 copies of every gene. Now, those 2 copies may be the same, or those 2 copies may be different, right? That make sense? Okay. Anyways, we'll continue with this. So here's the idea here. Okay, so let's just again assume eye color, single gene. We know it's not, but let's assume it is. And let's assume there are only 2 alleles for it, blue and green. And we know that that's not true, but we're assuming here, okay? Then P, which might represent the proportion of total alleles that are blue, plus Q, which is the proportion of total alleles that are green. Look at that. Man, you guys are on fire. It has to equal 1, right? Because there are only 2 different versions of that gene. And so those 2 proportions together better add up to the whole population, right? You're like, well, what about brown and hazel? Again, we're assuming that there are only 2 forms of this. So this is not the genetic structuring. This is the allele frequencies. This is the genetic structuring. Where now we say, how do those allele frequencies contribute to an individual's genotype? And so if we only have 2 different alleles, we have basically 3 different genotypes that are possible. We have what's called the homozygous dominant, where they have 2, in this case, where they have 2 blue alleles. Okay? We have the heterozygous individual that has 1 blue and 1 green, and then we have the homozygous recessive individual that has 2 green. And you're like, well, why'd you make blue dominant to green? We're just assuming here, right? We're playing a game. House rules. Okay? All right? This is the genetic structuring. When you make the connection between allele frequencies and genotypes, you've now talked about genetic structuring. Okay? So we're talking about what does this allele frequency actually mean at the population level in terms of what individuals actually have. Yeah, Chris.
How would you define allele?
So allele is just a particular form of the gene. In this case, we made 1 character, 1 gene, so the alleles are also the different traits.
Okay.
But if it's not 1 character, 1 gene, you would still have alleles, different versions of those genes. But in the case of human eye color, where there are 16 different genes that contribute it, we're talking about a lot of alleles, right? Some of those genes may have 100 different alleles.
Okay.
And so we're talking about a lot of potential variation. I don't know if you've ever been at an eye doctor that had a chart of the variation in human eye color, but it's pretty fascinating. Because most people have brown eyes, or something brown-like, right? Brown or brown-like eyes. And there's a good reason for that, because it helps you filter out enough sunlight that you don't get freckles on your retina that develop into, you know, skin cancer at the back of your eye, right? So, I don't know, newsflash, if you don't have brown eyes, you should probably wear sunglasses, because, you know, otherwise you're going to develop freckles on your retina that may... Anyways, sorry. Don't know if I, you know, ruined your life. If I did, I'm sorry. So, but anyways, if you see a chart where it just maps the variations in eye color, it's so fascinating, because it's really awkward to do this to actual people, right? It's just like, hang on for a second, I want to look at your eye up close, right? It's a little awkward, unless you're related to them, and even then, it can be pretty awkward. All right, so I want to point something out. Is there is an obvious connection between the genotype and the phenotype, right? The genotype is what 2 alleles do you actually have. The the phenotype is what is expressed. Okay? There's an obvious relationship between the particular genes that you have, the particular alleles that you have, and what you look like. Although what you look like adds more complexity to it. So I maintain this picture from the text, plus then it's fun to just like, look at the kitties, right? Forget all of the frustration of sitting here for 55 minutes with me, listening to me, and here's some kittens, right? But the phenotypes add to the complexity, so I don't want you to think phenotype when you hear genetic structuring. Even though there's an obvious connection between genotype and phenotype, right? Don't think phenotype. Think genotype, okay? And that's what I'm going to leave you with today. Don't think phenotype. Think genotype, all right? Have a wonderful conclusion to your Monday. Be safe. Make good decisions.